organizers for giving us the opportunity to share our data. We're going to be talking about the M2SR single replication live flu vaccine. The use of an M2 knockout was first described by Dr. Yoshi Kawaoka, a founder of FluGen, where he deleted the M2 gene from the wild-type M2 and resulted in the lack of M2 expression. And FluGen licensed this technology and has been developing it toward the clinic. Deleting the M2 gene results in a single replication. I guess this isn't pointing. In a single replication influenza vaccine that looks like a live virus, it can enter the cell and uncoat and undergo presenting the viral proteins, but because it doesn't have any M2, it can't make any progeny virus. So this replication deficient virus needs a production cell line that provides the M2 in trans, so the vaccine virus itself is complemented with the M2 protein. It looks like a normal virus. It's a 6-2 reassortant. The internal genes come from PR8, and the seasonal HA and NA genes are modified each season. This is the life cycle of the M2, of the influenza virus, and as you know, the M2 gene has ion channel activity. So the complemented virus does have this first part ion channel activity. It can enter the cell and uncoat, but since no new M2 is made, no new virus is released. So it's deficient in the second M2 assembly and morphogenesis phase. So therefore, there's no infectious virus. And this results in a live virus that can undergo the whole flu virus life cycle and generate mucosal immunity because it's administered through the natural route intranasally, induce the innate signaling like a normal flu virus, and then generate antibodies as well as T-cell responses to the internal genes. But again, there's no virus shedding because no new M2 is produced. And we've shown that this M2SR vaccine has, in animal models, both mice and ferrets, generated universal protection against multiple influenza subtypes. For example, the H1N1 vaccine has protected against H1N1 virus, H3N2, and H5N1. And what I'd like to do today is share bits of data from each one of these studies to show that we, with one vaccine, can generate these universal responses that are discussed in individual platform forms. So first, I'd like to talk about the H5N1 vaccine study. And these pointers aren't working. The mice were vaccinated with the homologous H5N1 M2SR, both in a prime-only and prime-boost vaccination regimen, and a heterologous group encoding the H1N1 HA M2SR was vaccinated in a prime and boost regimen. As a control, recombinant H5HA protein given intramuscularly in prime boost regimen. So the mice were challenged 20 weeks later. While we were waiting for the BSL-3 facility to come online, it inadvertently became a duration study as well. So after challenge, you can see that all the M2SR groups, both the single dose and the prime boost dose of H1N1 M2SR, the homologous group, protected all the mice, as did the H1N1 heterologous group. The recombinant H5HA protected only 40% of the mice, with the naive succumbing to infection. And when you look at the organs harvested after challenge, you see that the single dose and the prime boost dose with the homologous H5N1 M2SR controlled the challenge virus. No virus was recovered from any of the organs in the mice. The heterologous H1N1 group only had some virus in the lungs. It did control it from becoming a systemic infection, whereas the recombinant protein did let it go systemic, and the naive, of course, was systemic infection. When we then look at what types of responses are elicited by the M2SR vaccine, you see that in serum we do have IgG and IgA elicited by the H5N1 M2SR, as you do have the local lung wash having both IgG and IgA. And you also see that H1N1 by ELISA has cross-reactive antibodies against the H5HA. When you then look at what converts into functional HAI antibody, you see that the homologous H5N1, both prime only and prime boost, elicits high HI titers, and the challenge results was 100% protection. But the H1N1 M2SR that had cross-reactive ELISA titers does not elicit HAI antibodies, as you would expect, but did protect 100%. So in the next slide, we wanted to look at the mechanism of what that response was due to. And as on the left-hand corner, you can see the HA molecule. We in earlier talks discussed the stem antibodies as well as the variable HA head. So what we did was we ran out the recombinant H5, HA1, and HA2 proteins in a gel, and then transferred it in a Western and probed with the sera from the vaccinated mice, and see that the H5N1 M2SR recognizes both the H5, HA1, and HA2 regions as expected, but the H1N1 M2SR doesn't recognize the head, but does recognize the H5, HA stem, HA2 region. And you can see that the mock doesn't have any cross-reactivity against these proteins. So the M2SR does produce de novo HA antigens and elicit antibodies against the conserved stem region. Neuraminidase has also been mentioned as one of the universal cross-reactive antibodies that may provide protection, and you can see that the N1s are shared between H5N1 and H1N1, and we do elicit these cross-reactive neuraminidase antibodies. To demonstrate T-cell responses, we then moved from totally unrelated subtypes, an H1N1 M2SR vaccine, which is then challenged by IgH3N2 virus. And these mice survived the challenge, and what I'm going to show you is the T-cell responses from lung wash harvested day 4 and day 7 after challenge. And you can see both CD8 and CD4 peptide-specific responses that are similar in magnitude to the wild-type virus, a sublethal 10 PFU dose of control wild-type virus. So M2SR is able to elicit cross-reactive T cells to the internal genes like the NP and the polymerase genes. And as you know, these internal proteins are highly conserved amongst all subtypes, as well as from 1918 to today. And the recent H7N9, you see that the M158 antibody and other CD8 epitopes are fully conserved. When you look at the subtype of these T cells, you see that they are of the effector and effector memory subtype, and the magnitude, again, is similar to the wild-type virus that was used as a control. And this subtype of effector T cells were shown to result in reduced disease symptoms in the recent 2009 pandemic. We haven't talked about the elderly much today, but it's been shown by Janet McElhaney that the antibody response doesn't correlate with protection in the elderly, that the T cell responses are more indicative of protection. So the M2SR, particularly the granzyme B responses, as well as a high interferon gamma to IL-10 ratio. So as you can see, again, M2SR does have these responses that may serve in being protective in elderly. So we then moved on to the ferret model, and this study was sponsored by the NIH and conducted in Ted Ross's lab when he was in Pittsburgh. And it mimics the mice study described earlier, except in the ferret study, we also did add a prime-only H1N1 M2SR group. These are the HAI titers to the Vietnam 1203 virus, and you see that the homologous H5M2SR, both prime-only and prime-boost, are the only ones that elicit HAI titers in blue, and the heterologous H1N1 M2SR in green does not elicit HAI titers against H5. Okay, after challenge, you see that the homologous H5M2SR prime boost group protected 100% of the ferrets, and the prime-only single dose protected 2/3 of the animals, and the naive succumbed to infection. When you look at the survival results for the heterologous group, the prime boost dose of H1N1 also protected 100% of the ferrets, and the H1N1 M2SR protected 2/3 of the animals. When you look at the clinical symptoms for the after challenge, you see that the prime-only, both the H5 and H1M2SR, did not have any clinical symptoms, clinical scores in the ferrets, and the single-dose homologous one in black had less clinical symptoms than the H1M2SR prime-only in blue, and the naive, of course, exhibited more clinical symptoms. And the M2SR also controls shedding of the challenge virus. In red, you have the mock PBS group where you have virus titer in the nasal wash. The H5N1 prime only and the H1N1 prime only also had virus in the nasal washes, whereas the prime boost groups control the challenge virus, and there's no virus in the challenge in the nasal wash from the prime boost groups. And when you look at in the histopath scores from the lungs harvested from the ferrets, you see that there's no virus from the prime boost groups, and the prime only had low histopath scores in the lung and very low virus titer recovered from the lung. So as you know, there's another live vaccine that is commercially available, Flumist, and we wanted to compare M2SR to see how that compares in animal models. As you know, Flumist is very, very good in children and naive populations, but it's less effective in adults, presumably due to the role of the pre-existing immunity. So we wanted to do some mice studies and compare some of our responses. As discussed earlier, the HAI titer is the primary flu measurement for licensure. So in mice experiments, we see that M2SR is able to elicit higher HAI titers than Flumist. And when we do an intrasubtypic challenge with a related Aichi virus, you see that M2SR does protect better than Flumist. And when you do a dose titration with a fully heterologous challenge, so an H3 vaccine challenged by an H1, or H1 vaccine challenged by an H3, you see that the dose response, the M2SR being in blue and the Flumist in red, that there is more of a separation as you decrease the dose. So that made us want to look at more the molecular basis of the differences of the two vaccine viruses. So we looked at them in vitro in cell culture and did a growth curve. This is in M2CK cells that are providing the M2 protein in trans. And so our first look at the growth curve, and you see that at 24 hours in the day 1 post-infection, that M2SR mimics wild type, and there's a greater than 2 log difference between the Flumis vaccine virus and wild type and M2SR. So we then looked at that more in non-permissive cells, A549 cells, and focused on the 24-hour time point and looked by FACS at infected cells stained for NP. And you see that antigen production or protein production, again, M2SR mimics the wild type and Flumis is delayed. So we think that maybe this early protein production kinetics may overcome the pre-existing immunity, and having a potent generation of antigen may help. So this is taken by a review from 2002, Brian Murphy's work on all the challenge studies, and Flumis has been shown in adults to not replicate as well as it does in children. Here, if we differentiate between seronegative and immunologically naive, seronegative meaning there's no HAI titer, you see that wild-type viruses can replicate to 10 to the 5th. In adults and elderly adults, Flumis cannot. But in immunologically naive, meaning non-antibody or non-neutralizing response is totally naive, Flumis does replicate up to 12 days and is able to generate the protective responses. So it's the pre-existing immunity, non-HAI-based immunity, that's affecting the virus. And that's what we wanted to check, our surviving mice. We had some survivors that we kept around that have experience from H1 vaccination, H3 challenge, and we kept them around. By then they were 18 months old, and we wanted to see if we gave them a new closely related M2SR, could they elicit a response in the presence of all this pre-existing immunity. So we vaccinated with the BRIZ 2007 M2SR, and they have cross-reactive IgE antibodies in them, and we see that those were boosted twofold, but new responses to the Brisbane 2007 were elicited a fourfold response by ELISA. And then looking at it for even more closer HAs, meaning 1 season apart, because IgE and Brisbane are 46 years apart, you see that if you vaccinate with TIV containing the Perth antigen and then come back with M2SR Briz, so 1 season apart, that we can boost the antibody titers even when it's closely related HAs. And then just to show that it's not just a virus particle being processed, that it does actually undergo an infection and uncoat and make the new antigens, we show that the T-cell responses are increased after IgE and vaccine, so priming with a virus and then coming back with the vaccine using an NP147 tetramer. So it's not just the virus particle being processed, that it is undergoing infection and making the internal proteins. So in summary, we've shown that the M2SR does generate universal responses. We've shown multiple subtypes being protective against each other. It's not just one subtype. The H3N2 also protects against H1N1. And in one vaccine, we do have the broad spectrum universal responses that are individually considered universal platforms, the T cells, the stem antibodies, as well as mucosal responses. And we are in a process of going to clinic, Q1 of 2016, and hope this pans out in the humans. I'd like to acknowledge all our collaborators, and we'll take any questions. Thank you very much.
Thank you for the great talk. So now it's open for discussion. So are there any audience have the... Yes, go ahead, please.
Have you ever, excuse me, have you ever looked at protection against flu B, or are you looking at all at flu B versions of your M2 deletion?
So we are in development with the flu B version. The flu B version has a different M2SR backbone, a BM2SR. The M2 there is not overlapping in the M gene and the B sequence, but there is one coming across. Yoshi Kawoka has also published on the M2 knockout version, so we are developing that, and that will be part of the quadrivalent version.
Yes, go ahead.
Why are you focused so much on H1 rather than H3?
It must have come across as a focus of H1, but we're doing both. Converse, we also have H3 and ferret data, and also, no, we're not focusing on H1. It just seemed like that, I think.
Nobody? So that, oh, please.
Do you do any genetic stability for the virus?
Yes, as part of our preclinical package for the FDA, we have been passaging our vaccine virus. In vitro, it's gone 20 passages without any revertants. We've also done mouse-to-mouse passage to select for revertants, and it's genetically stable. We haven't observed any.
So for the seasonal strain, like how many strains you have done? I mean, how many seasonal strains you have done for this platform?
Oh, maybe 3 or 4 H3s and that many H1s.